DNA nucleotides. And so far, right, those are all the same steps that we've done with leading strand replication. Now here's where the real difference comes. The problem is, DNA polymerase III, even after, and I'm going to erase this primer really quickly just for the sake of demonstration. So what polymerase I has done, right, is it's applied DNA nucleotides in the place of those RNA nucleotides. When DNA polymerase III gets to this point where it reaches that first Okazaki fragment, it can't create a bond between that last nucleotide there and the first nucleotide in the first Okazaki fragment. So there is a gap right here. That's when the last enzyme comes in. And the last enzyme, which is called ligase, creates a covalent bond between that nucleotide and that nucleotide. Okay, so this is step number 4. Ligase creates covalent bond between Okazaki fragments. Okay, so let's think about this from kind of a macroscopic level. We know that the replication direction of each Okazaki fragment is from 3' to 5', right? But what's the overall direction of growth of that lagging strand's complementary strand?
Yeah.
The opposite way, right? I mean, overall, the DNA is still being replicated in that direction because we have an Okazaki fragment, then an Okazaki fragment, then another Okazaki fragment, and it keeps growing in discrete units from the left to the right, following helicase. Okay? The top leading strand replication goes from the left to the right continuously. Lagging strand goes from the left to the right discontinuously. You okay with that?
So wouldn't that take like just a little longer?
It's certainly not as efficient a process, yeah, for sure. Okay, what questions do you have about leading and lagging strand replication? Okay, so on the exam, you can expect questions like, which of the following enzymes accomplishes something? Applies DNA nucleotides to the complementary strand, growing complementary strand. That's going to be DNA polymerase III. Which of the following enzymes attaches Okazaki fragments with a covalent bond? That's going to be ligase. Things like that. How do you guys feel about this diagram? Do you think that you could replicate this on an exam? I would be prepared to replicate that on an exam. And did you have a question?
Yeah, what does that little circle stand for? That's ligase?
This right here?
Yeah.
So that shows that there isn't a covalent bond between those two Okazaki fragments. But yeah, that's where ligase will be active. It's going to connect that to that. Now, yeah, feel free to take a picture of it if you'd like. Now, here's the million-dollar question. There is a particular type of covalent bond that exists between nucleotides. And it's actually the type of bond that ligase creates. Do you remember what that bond was called way back from chapter 4? Yeah, a phosphodiester bond. Yeah. So that phosphodiester bond is a product of dehydration synthesis, right? Because what's happening is ligase is taking an OH from one group and an H from another group, creating a water, opening up a covalent bond space, and those two things are stuck together. Okay, good. Questions? All right, well, let's get back to the PowerPoint. Okay, so this is going to go from leading strand replication all the way into lagging strand replication. So the first thing to happen with leading strand replication is that helicase comes along, right? And it unwinds and unzips the DNA molecule. And that's going to happen throughout the chromosomes that we're replicating, right? There are going to be different points at which different helicase proteins bind and unwind and unzip. And essentially what that helicase is doing is it's disrupting the hydrogen bonds that exist between complementary strands of DNA. Now, to keep those two complementary strands separate from one another, we're applying single-strand stabilizing proteins so that those hydrogen bonds don't spontaneously reform. And that's what you see right here. Helicase is unwinding and unzipping, and we're applying single-strand stabilizing proteins to make sure that those two complementary strands stay apart. Now, when helicase reads it, right, it's reading along the leading strand from the 3' to the 5' direction. Okay. Then what we need to do is we need to make it so that DNA polymerase III can actually attach to the template strand and start creating a new complementary strand. And the way that we accomplish that is by having primase come along and apply an RNA primer. And please remember that RNA primer really just acts as like a landing pad or a landing structure for DNA polymerase III. Okay, so it applies a primer, but that primer consists of RNA nucleotides, right? After that RNA primer is applied, the DNA polymerase can come along and it can land and it can start creating a complementary strand to its template. And it does that by applying the appropriate DNA nucleotides. After that's been accomplished, and that's what we see happening right here, so we have primase that's created a primer, and the DNA polymerase III that's on the leading strand beginning to grow out right behind helicase. And it just continues to do that. We then need to replace that RNA primer with DNA nucleotides. And DNA polymerase I accomplishes that. And then that's leading strand replication, really. Lagging strand replication is a little bit different. It occurs in discrete fragments called Okazaki fragments. And functionally, it's the same thing. At discrete points, we're going to have the application of an RNA primer. DNA polymerase III is going to grow that out. DNA polymerase I is going to replace the primer. And then primase is going to jump forward, create a new primer, and then DNA polymerase III is going to grow that out to the first Okazaki fragment, right? And then we have that final enzyme that comes along and creates a covalent bond between Okazaki fragments, and that's called ligase.
So Dr. Tanner, why isn't there like the SSSP in the lagging strand?
There can be,
Okay.
but really, if you're applying single-strand stabilizing proteins here, you don't need to worry about applying them there because those,
They're already stable.
yeah, they can't reform. Okay. Questions? All right. So, unfortunately, mistakes on occasion happen during replication, and that makes some intuitive sense, right? Because we know that the DNA molecule is really, really long, consisting of billions of nucleotides. Sometimes a single nucleotide can be replaced by another nucleotide. Like purines can be flip-flopped accidentally. So instead of an A, we get a G. Or instead of a T, we get a C. That exchange, or that mistake in replication, is by definition a mutation. A mutation is any change to our DNA. Now,